Welcome to today's lesson on enzymes. In this session, we will explore the nature of enzyme molecules, as well as how their activity is influenced by both temperature and pH changes. In our lesson today, we will explore in turn how temperature and pH impact enzyme activity. So far, you have learned about the importance of enzymes in speeding up biochemical reactions without being consumed in the process. You also know that enzymes are highly specific, meaning each enzyme only catalyzes a specific type of reaction or acts on a particular substrate. Let's get started. Temperature significantly impacts on enzyme activity. At low temperatures, enzymes and substrates have very little kinetic energy. This means that they move slowly and have fewer collisions with each other, which results in a slower rate of reaction. However, as the temperature rises, both the enzymes and substrates gain kinetic energy, move faster, and collide with each other more often. This increases the rate of reaction up to an optimal temperature. Every enzyme has an optimal temperature at which it functions most efficiently, and therefore causes the fastest rate of reaction. For most human enzymes, this temperature is around 37 degrees Celsius, which is normal body temperature. Beyond the optimum temperature, the bonds holding the enzymes into its unique shape will break, causing it to denature. When an enzyme denatures, it alters the active site, so the substrate no longer fits, and the enzyme activity therefore declines sharply. We can also visualize the effect of temperature on an enzyme by using a graph that shows the rate of reaction against the temperature. Let's take a closer look at this. As the temperature increases, enzymes and substrates have more kinetic energy, more collisions, and therefore a faster rate of reaction up until an optimum temperature. As we mentioned, this is about 37 degrees in the human body. At this optimum temperature, the rate of reaction reaches its peak. After this optimum temperature, the enzymes begin to denature, meaning that they can no longer bind to the substrate, and therefore the rate of reaction decreases. Now let's look at how pH levels affect enzyme activity. When the pH is too low and strongly acidic, or too high and strongly alkaline, it can disrupt the bonds that hold an enzyme's structure together. This disruption can cause the enzyme to denature, changing the active site so that the substrate can no longer bind, and leading to a decreased reaction rate. Each enzyme has a specific pH level at which it works best. For many enzymes, this is around pH 7, which is considered pH neutral. However, there are exceptions. For instance, pepsin, an enzyme that digests proteins in the stomach, works best in acidic conditions at around pH 2. This matches the stomach's environment. You can visualize the effects of pH on enzyme activity on a graph. To do this, we plot the enzyme activity against the pH. The graph usually shows a peak at the enzyme's optimum pH, with a steep decrease on either side, as the enzymes become more denatured in acidic or alkaline conditions. Different enzymes have different optima, so will peak at different pHs of the graph. For example, pepsin is an enzyme that works in the stomach to catalyze the breakdown of proteins. Remember that the stomach produces hydrochloric acid to kill bacteria, therefore it is an acidic environment. This is why pepsin's optimum pH is lower or more acidic than most other enzymes. Before moving on, let's consider the historical background of pepsin as an extension task. The study of digestive enzymes began with the discovery of pepsin by Theodor Schwann in 1836. Let's now take a closer look at this discovery.
In 1836, during my studies of the digestive system, I discovered pepsin. This discovery marked the first time an enzyme was ever isolated, a significant breakthrough in the field of biochemistry. it became clear that pepsin played a crucial role in breaking down food within the stomach, a realization that opened the door to a deeper understanding of digestive processes. This finding not only advanced our knowledge of how food is broken down, but also laid the groundwork for future research into the vital functions of enzymes in the body.
In today's lesson, we focused on how enzyme activity is influenced by temperature, as well as by pH changes. We first examined how temperature can affect enzyme activity by changing the kinetic energy of both enzymes and substrates, and therefore the rate of collisions. Each enzyme has an optimal temperature where its reactions are most efficient. Temperature can also cause enzymes to denature when it is too high. Next, we explored the potential impacts of pH on enzyme activity. pH can cause enzymes to denature when it is too far away from the enzyme's optimum. Most enzymes have an optimum pH of around 7, also termed neutral. However, there are many examples that do not have this optimum pH, such as pepsin.
Watch the rest of the GCSE biology course at Sophos Education.